Every single second, billions of chemical reactions are happening inside your body. But here's the twist: without one special type of molecule, life as we know it would simply stop. These molecular masterminds are called enzymes. And today, we are going beyond the basics into the deep science that makes enzymes the ultimate architects of life's chemistry. Enzymes are biological catalysts, mostly proteins, and in rare cases are in a molecule called ribozymes. They speed up reactions not by magic but by lowering the activation energy, the invisible barrier that slows reaction down. Without them, the chemistry of life would be so slow that you wouldn't be alive to hear this sentence. Every enzyme has an active site, a microscopic pocket perfectly shaped for its target molecule, the substrate. Here, precision chemistry happens. Non-covalent forces like hydrogen bonds, ionic interaction, and hydrophobic effects guide the substrate into place. And sometimes amino acids like serine, histidine, or aspartate steps in directly to break or form the bond. Enzymes work in several clever ways. They bring molecules together, the perfect orientation. They use acid base chemistry to transfer protons. They form temporary covalent bonds to push reaction forward, and some even recruits metal ions like zinc or magnesium to stabilize the charges. The result: a reaction that's millions of times faster than it would ever be on its own. Scientists study enzyme speed using the Michaelis-Menten equation. The Km value tells us how much substrate is needed for half max a year of enzyme affinity. High Km, more of the distant acquaintance. This math lets us predict exactly how enzymes behave in the chaos of living cell. But enzymes aren't always on full power. They can be switched on or off through allosteric regulations, feedback inhibition, or even chemical modifications like phosphorylation. Some are born inactive or activated later, like digestive enzymes that would destroy your pancreas if they work too early. Next, we have cofactors and coenzymes. Cofactors are inorganic ions like zinc and magnesium. Coenzymes are organic molecules derived from vitamins like NAD positive or niacin, FAD from riboflavin. Prosthetic group: the tightly bound cofactor or coenzyme is known as prosthetic group, like heme in cytochromes. Six major classes of enzymes are here: oxidoreductases, electrons transfer reaction; transferases, group transfer reaction; hydrolases, as the name represents hydrolytic bond cleavage; lyases, addition removal of group to double bond without hydrolysis; isomerases, intermolecular rearrangement; ligases, bond formation with ATP hydrolysis. Enzyme inhibition advanced view. Competitive inhibition increases Km, Kr, Vmax. Vmax remains unchanged. Non-competitive inhibition decreases Vmax while Km and Kr. Uncompetitive inhibition decreases both Km, Kr and Vmax. Irreversible inhibition, covalent modification of enzymes, e.g. aspirin, acetylates, COX enzyme. Enzymes aren't just in biology textbooks. They diagnose heart attacks, treat cancer, remove stains from your clothes, and even copy DNA in forensic labs. Without them, medicine, industry, and biotechnology would be crippled. Food industry: amylases in baking, rennin in cheese making, detergent; proteases for stain removal; molecular biology: DNA polymerases in PCR, restriction endonucleases in genetic engineering. From the tiniest pocket in the proteins to the vast network of life's chemistry, enzymes are the invisible engineers keeping you alive. Right now and next time you breathe, blink, or even think, you can thank an enzyme for making it happen. If you enjoyed this deep dive into the enzyme, hit the like button, subscribe for more biochemistry adventure, and drop your next topic request to the comment. Maybe your idea will be the next deep science story we explore.